the first thing that we have to do is actually take the Dura off so that we can get down and see those internal structures. So I want to show you guys what's going to be the easiest way to cut through your sample. If you start on this portion here where you actually can see the portion of your olfactory bulbs that are actually exposed there, that's the easiest place to kind of work your scissors in and start cutting away at the Dura. So you'll notice here you can kind of put your scissors in here and start cutting this way. Flip the brain over and cut the superior portion here so you can cut it all along the length there. And that's the easiest way to get started cutting your Dura. And you actually can look at it here, you can see how it starts to pull away. The thing that you want to be careful about, there's two areas where the meninges are actually attached. The first one is going to be down inside your longitudinal fissure. It actually dips down inside there. So you're going to have to pull it out in between the two cerebral hemispheres. And then also down inside your transverse fissure, it's going to dip down in there as well. So you're going to have to kind of go down in the fissure and cut that tissue away in order to be able to remove it. The other area where it's going to be attached is going to be associated with your cranial nerves. When you get ready to remove the Dura, if you're not careful with it, you actually will pull off the cranial nerves. So if you notice here when I kind of cut along the side, if I lift the meninges up here, you'll be able to see the cranial nerves that are actually extending through the meningeal layer here. So if we cut right across here, see this portion right in there? That's actually a portion of the optic nerve that is extending through the meninges. You'll also find several of your smaller, like cranial nerves 3, 4. Those are going to be small nerves, and when you pull the meninges away, you'll be able to see that they're extending through the meninges. So see this tiny little structure right down in there. I don't know how well you guys can do it on the screen. There's a little structure right down in there that's actually running through the meninges. You want to take your scissors down there and you actually want to cut it away. If you just pull the meninges away, it's going to pull it off at the root, and you're not going to be able to see where it was actually attached to the brain. So you want to make sure when you get to this inferior portion that you just kind of lift the meningeal layer up. Anything that you see that's attached to the meninges, cut it so that there's a little stump of it left so that you can actually see it and identify your cranial nerves. Okay? So go ahead and start working on your meninges, get all of that